Hi guys, good evening. In this live session today, we'll discuss week 1 lectures of Demystifying the Brain course. I'm Deepak Raya from Center for Neuroscience, IISc Bangalore. Let's start. What are these live sessions about? It's a review of week's lectures and we'll add some complementary to the topics covered. Doubt clearing sessions. If you have doubts, you can ask me and we can discuss about them. And I'll share some useful and relevant resources that might be useful for your preparation. The timings of this live sessions are every Saturday 5 p.m. to 7 p.m. So, what happened in this week 1? We have discussed some important events in the history of neuroscience, which led to few fundamental principles or basis for the entire field of neuroscience. Then we'll discuss about basic neuroanatomy as well as microanatomy, that is, what types of cells are present in the brain and how they are connected to each other, etc. We also dealt a bit with some of the principles of neural design, for example, the save wire principle, and also we dealt with what are the implications of brain size and its placement, how are the connections present, what type of connections are present, some evolutionary insights like how the brain developed in different organisms. And finally, we ended with some overview or a big picture of neuron signaling. So, some of the important events in the history of neuroscience. In the 19th century, Ramoni Kahl and Golgi came up with this thing called neuron doctrine. When they stained the neurons of a brain tissue, they found that the nervous system is made up of individual cells called neurons, and it's not a continuum. Each neuron is connected via a gap called synapse. It's a very tiny gap around around the order of nanometers. And this work won a Nobel Prize in 1906. And in 18th century, Luigi Galvani found the electrical property of these cells. So when he stimulated a neuron or a nervous cell in a frog leg, he was able to see a response in that dead frog leg. This paved the way to a field called electrophysiology. Later on, Hans Berger was able to record brain signals by placing electrodes on the scalp, which is called electroencephalogram. But electroencephalogram is very coarse in nature because you cannot find what is a source that is giving particular signal. So to get more finer view of neuron signaling, Hodgkin and Huxley were able to record from a giant squid axon. They were able to record membrane potentials and come up with mathematical and electrical properties of this membrane potentials, which is an important property for action potential generation and etc. And this work by Hodgkin and Huxley won a Nobel Prize in 1963. Furthermore, in early 20th century, Paul Ehrlich, John Langley found something called magic bullet and receptors, where you could activate particular cell by a chemical substance when it gets attached to that particular receptor. Later on, Otto Loewy's experiment showed that there is a chemical synapse between the neurons. The synapse, the gap between two neurons, is activated via chemicals. These chemicals are called neurotransmitters, and whenever the action potential reaches in the presynaptic neuron, it just releases some chemical substances which are being received by the postsynaptic neuron and then gives the response. So all these important events in the history of neuroscience led to a full view, microscale view of the nervous system. So our entire nervous system is made up of these cells called neurons. Each neuron, let's say this is the synapse at this point, the red ones you see here, and this is called presynaptic neuron, and this is called postsynaptic neuron, right? So the signal generated in this presynaptic cell passes down the axon and received on the next postsynaptic neuron. As the action potential reaches the end of this neuron, the events enable some of the vesicles are small sacs of chemical molecules. These action potential informs these vesicles to fuse with this wall of the synapse and releases the neurotransmitters to the next neuron, which binds to the receptors here and then propagates the signal to the next neuron. So this is how the whole neuron, the basic information processing and basic units work together in your nervous system. Let's move on to discuss why do we need a brain. So neuroscience deals with reverse engineering the brain. You want to understand the brain by using reverse engineering. So what are the three main questions that you ask for this is, what does it do, what are its specifications, and what is the environmental context. For example, if you take E. coli, the bacteria, it succeeds without a brain because it doesn't need much of the processing. It just needs to find the food and go near that. So if it can sense the chemical, if it can sense the food gradient, concentration gradient, it can just go there wherever there is high concentration of food. It goes and lives there, and it can succeed without any brain. Then going down the food chain, you have paramecium choritum. It is brainless but needs faster signaling. Then coming to C. elegans. C. elegans has a nervous system with exactly 302 neurons. So which has properties such as it is able to find, ingest and digest bacteria, recall favorable temperatures and bacterial concentration. So this aspect of recall and storage needs the brain. Small organisms do learn and remember to just match their own life context. Now, with progressing down the food chain, the computational complexity increases because it has to have more capacity to inhabit richer environments. And different organisms have different types of different sizes of nervous systems and number of cells, etc., because they want to pay only for what they use. So this is the main principle that drives. And so what fundamental, okay, a question for design principle of the nervous system is, what fundamental purpose does it serve, and at what cost to the organism, right? So what's the core task of a brain? Does anyone know what's the core task of a brain? Yes, exactly. So it is used for homeostatic regulation. What I mean by homeostatic regulation, wait for a parameter to deviate from a set point, detect the deviation, and correct the error via feedback and control the process. But apart from this, the main task of the brain, like for that matter, any organ in the body works on this principle of homeostatic regulation. But the extra function that comes up with the brain is it can do something called as anticipatory regulation. So what is anticipatory regulation? Is responding to the needs. Like you have to predict it before, even before the need arises, and then serve it, anticipate the needs, satisfy them even before they arrive. And what are advantages of this anticipatory regulation is respond capacity to fluctuations in demand, and they can allot the things according to their needs even before the crisis arises. This way, and also they are able to minimize the errors. Few examples of anticipatory regulation and behavior. For example, you can reduce the demand for physiological cooling, that is through sweating, which consumes a lot of resources such as sodium chloride and water. In order to be away from that, your brain tells you to find a shade so that you can reduce the need for physiological cooling. And also, another way to put this is you have a memory of heat stroke, and therefore your brain signals some of the anticipatory measures to find a shade or uh, to find shade and relax. Therefore, you have memory in for most persistent dangers, and these few memories even exist beyond generations. For example, if you take monkeys, they have a fear of snakes. This is a kind of fear is also a kind of anticipatory regulation. Then people in the society has more or higher amount of reserve for acute observations than compared to an individual, and therefore we live in groups and learn from groups. So this groups and reacting how the group responds is also kind of anticipatory regulation, where and also some of the emotions arise because you must continuously sense the social structure and behave yourself. So this way the social structures are also kind of anticipatory regulation, and your brain responds to this via different emotions, language, speech, etc. Then, and also, you're tuned to environmental regularities. For example, circadian rhythm and seasonal rhythms. Circadian rhythm is nothing but your sleep and wake cycle and how it progresses over the day. And seasonal rhythms are basically the best seasons to forage for food or for mating, etc. So our brain is also tuned to this environmental regularities, and these are all few examples of anticipatory regulation. And also you must have uh, some estimate to know whether you are living optimally. For that, whenever you reach a goal or something, you feel good, and whenever you don't, you feel bad. This kind of regulation is also enabled by the brain. So yeah, let's deal few terms about neuroanatomy. So before going into neuroanatomy, just discuss few anatomy terms which are very useful across all domains. So here you see a human body with different planes here, and each plane has its own name. Let's say the plane A, which is cutting the body to halves, left and right half. So this is called sagittal plane. So have you heard of uh, this sign called star sign called Sagittarius? It's like a bow and arrow thing. It's like slicing something into two halves. So here you're slicing the human body into two halves, that is left and right half, and this plane that is called sagittal plane. Then there is a plane which cuts the body into top and bottom halves, which is called transverse plane. Transverse. And there is a plane which cuts the body into front half and back half, which is called coronal plane. And the axis passing through the center of your body is called longitudinal axis. Along the body there is an axis which is called longitudinal axis. So this D is called longitudinal axis. This C is called transverse plane. Left and right halves is called sagittal plane. And sagittal, transverse, coronal. What is the coronal plane? Which shifts your body into two halves, right? Sorry, there was an interruption in the current. Yes. Let's continue. And there are few other terms apart from this important planes. So just to recapitulate, sagittal is something that cuts your body into two halves, left and right. Coronal plane is something that cuts your body into front and back half, and transverse plane is something that cuts your body into top and bottom halves, and an axis passing through the center of your body is called longitudinal axis. Apart from this, we have proximal is towards, distal is away, like something going like this is towards, something going away is distal, away from some center. Then medial is towards the middle of your body. Away from middle of your body is called lateral. Then towards the front is called anterior, towards the back is called posterior. These are the few important terms in any anatomy that you find in the literature. Now coming specifically to the brain, apart from these planes that we have learned, we'll first start with some regions of the brain. So brain is divided into these regions in different colors here. One is the frontal lobe, the blue colored region, then a parietal lobe, occipital lobe at the back, temporal lobe towards the ear, and the cerebellum towards backside and under the occipital lobe. These are the main lobes of your brain. And then there are few important gyri, which are these dips here. So this long dip you see here is like just before the central, so it is called precentral sulcus, this part. Central sulcus is this part. Sylvian fissure is that separates parietal lobe from temporal lobe. Parietal lobe from different lobes. Then you have occipital lobe. And you have postcentral gyrus. So this sulcus is the dip, and gyri is the folds. These folds. Yes. And you see here around the central sulcus there is postcentral sulcus, precentral postcentral gyrus, precentral gyrus, and this forms the postcentral forms the somatosensory cortex, and precentral forms the motor cortex, primary motor cortex, and on towards the temporal lobe is your auditory cortex, and occipital lobe is the visual cortex, which is involved in visual processing. Okay, is this clear about few terms in neuroanatomy, basically the brain regions? Okay, now let's move on to the nervous system. So nervous system is made up of two things. We have discussed a bit about brain's anatomy. Apart from brain, you have a spinal cord, which is also part of nervous system. So you have a central nervous system and peripheral nervous system. Central nervous system contains two parts, brain and the spinal cord. Peripheral nervous system contains the nerves passing from the brain and spinal cord to the other parts of the body. All these nerves, this forms the peripheral nervous system. So some terms are just given different names depending on where it is, whether it is in the central nervous system or peripheral nervous system. So whatever is ganglion called in peripheral nervous system, it's a group of cells, is called as nucleus in the brain and spinal cord. Nerves are called as tracts in the brain. So this way there are some terminology differences depending on where a particular anatomical structure is located, like either in the central nervous system or peripheral nervous system. This is a big picture view of anatomy and anatomy terms. Now you see a coronal plane here, which divides the brain into two halves, front and back half, and a slice here is taken, and there is a transverse plane to the spinal cord. So what you see here is, this is a spinal cord and this is in the brain. You see this has gray matter and internal side is white matter. Here it just reverses in the case of spinal cord. You have outer side has white matter, inner side has gray matter. So what is gray matter? Okay, first discuss what is white matter. So white matter is made up of these axons. These are the connections between neuron to neuron and neuron to other neurons in the other regions. And these axons are myelinated, that is covered with the wrap of a fat tissue called myelin sheath, and this makes majority of this connections are made up of these axons and which are covered with this myelin sheath. So you see that the appearance of this is like a white matter, whereas this have more of cells than connections, therefore it looks grayish. And on the other hand, the cells are located here and the axons move to the peripheral system, and therefore the cell bodies here connect with each other, connect with other regions, etc., connect to the spinal cord. From spinal cord, these cells project onto peripheral nerves, and therefore there is this white matter and gray matter in brain and spinal cord. Coming to the microarchitecture, that is the basic unit of a nervous system, which is a cell. So these cells are electrically active and named as neurons. So you have a cell body with projections like this, and on the side of the body you have These projections called dendrites, and a longer projection called axon, which passes the signal to the next neuron, which is covered with this myelinated fat based tissue called myelin sheath, and this helps in faster propagation of action potentials. Then you have some terminal ends called terminal butons. They look like buttons and therefore named as butons. These connect onto the dendrites of the next neuron called postsynaptic neuron. This is called presynaptic neuron. So dendrites receive message from other cells. They integrate at the soma and pass on. When they create an action potential, pass on through the axon, going reaching the ends of this neuron called terminal butons. From there, they propagate the signal, they communicate the signal to the next neuron. But this is only type of cell that we discussed in the brain. But there are other types of cells present in the brain called astrocytes and dendrocytes. These are all types of glial cells which helps in nourishment and also in signaling of the neurons. So this oligodendrocytes, a type of glial cells, give rise to this myelin sheath, and these astrocytes help in immune response and nourishing the neuron. Yeah. And within the neurons, there is a far diversity. Like not every neuron looks same. Uh, this is called a pyramidal neuron. This is called a Purkinje neuron. So different neurons have different types of branches and arbors, and this way you have high cellular diversity in the nervous system itself. Within the neuron, as well as you have other types of cells such as oligodendrocytes, astrocytes, and other types of glial cells. So this is about microarchitecture or microscopic view of nervous system, and we have dealt with macroscopic, that is the general neuroanatomy. So this is about anatomy of the cells. Now let's shift our gaze towards some of the constraints for neural design. So first thing is, with rise in information rates, the cost also rise disproportionately. That is, in order to give information, more amount of information, your neurons has to spike at higher rates. But higher rates also comes with a cost, because it has to send it to other neurons as fast as possible and without any delays. So to transmitting more information requires higher spike rates. With increase in spike rate, axon diameter also increases, because with increase in axon diameter, the propagation speed increases, as we have seen that the myelin sheath improves the speed of propagation. And with axon diameter, rise in axon diameter, volume and energy also rise. So wanting in more information rates create costs at different levels. So your brain has to optimize its structure in order to optimally use these variables, like the information rates, energy and volume requirement. So it's basically, you have heard in electronics called power, speed and time, power, space and time. So you have to optimize this variable such a way that your circuit optimally uses these resources. Okay, and similarly that applies to the brain. So one of the key to design is send only information that is needed. You don't want to increase your information rates disproportionately. So send only what is needed and as slowly as possible. So at the same time you want a fast propagation, that is true, but it is like at the faster level itself as slowly as possible, so that your information rates are optimal and do not increase the cost disproportionately. And another constraint for neural design is a 100 millivolt spike takes less amount of energy compared to a 10 millivolt spike. So it is cheaper to compute with smaller currents or smaller voltages, and therefore another key to the design is minimize energy per bit of information by computing in the finest level possible, right? Then you have diversity of neurons. Someone can ask a question that why do we need so much diversity in the neurons? So if you can divide the information processing into different types of neurons, and therefore which helps the neurons to send only information that is needed. As you have seen in the first thing here, send only information that is needed. So this diversity helps in this thing. Parallelly send information only that is needed. And then coming to the wiring efficiency. So in least space and time energy, you want a best performance. This is what you want in circuits also, in electronic circuits. And this comes with various principles, design principles such as save wire principle. You want to place your brain or the connections of the brain such that it minimizes the total wire length compared to any other wire length in any other configuration possible. So this is one of the reason for why your brain is located in your head, because there are far more connections to your sensory organs on your face, such as your tongue, lips, eyes, etc. And therefore there is high density towards your head compared to other areas, and therefore your brain is placed there in order to minimize the total wire. And also electrical resistance increases with the increase in distance. Therefore there is also, in order to double the conduction distance and half the conduction delay, the volume of the cell has to be quadrupled, and at the same time there is a constraint on volume. So this way your brain has to, like, the design has to optimize how your brain and nervous system has to be organized. And these are the few principles apart from save wire principle that we discussed in the lectures. And also the neural system is designed for learning, right? In the sense, what I mean by that is, it is better to store the information wherever you encode rather than storing in a different place. Therefore the memories are stored in the synapses by making some physiological changes at the level of synapse. And this way, like, these are the few principles of neural design. We didn't go into the details, but these are the few principles of this. So now coming to neuron signaling. So neurons are electrically active. So at rest they have a negative potential which is maintained at minus 70 millivolts, maintained through various mechanisms. Uh, the main one is called sodium potassium pump that we'll deal with in the next class. Uh, so the cell is resting at minus 70 millivolts, and as it receives current, more and more current, the voltage rises, and when once it reaches a threshold voltage, it just increases rapidly until a final point like 30 millivolts. This stage is called depolarization, and once it reaches the maximum possible level, it slowly falls, which is repolarization, and there is small amount of decrease in hyperpolarization, and then comes back to resting membrane voltage. So this is how a typical action potential looks. Okay. Action potential looks. So once it reaches threshold, it just increases to the maximum possible voltage. Here it is plus 30 millivolts, and then slowly other mechanism starts, which repolarizes the neuron back to rest. And other thing here is, 1 second. Okay, something is not going. So here you see, I wanted to show you about the use of this myelin sheath. So this axon is covered by this myelin sheath, whereas this is not covered. You see, the signal is faster in the case of myelinated cell compared to unmyelinated cell. It propagates very slowly, whereas here it propagates very fast. This is because the signal is being regenerated at every gap here instead of propagating. That way, it reduces the dispersity of the action potential as well as the amplitude. So this helps in regenerating and fast propagating compared to without unmyelinated cell. So we'll discuss details about this. This is called saltatory conduction. Saltatory conduction. Okay. Okay. Now, some of the insights into some neuronal signaling after seeing the action potential. So, there are many currents coming into the cell body through the dendrites, and this current might increase the voltage of the neuron to some extent, and once it reaches the threshold, it just fires an action potential. But since the neuron is receiving multiple signatures, and it should reach a threshold to fire an action potential. So if it doesn't reach, it doesn't fire any potential, but it can also happen. This is called temporal summation, where action potentials close in time could help the neuron to cross a threshold. So the first first pulse just excited the neuron a little bit. It is almost at that state, and at the same time another impulse arrives and just rises and crosses the threshold. And once it crosses the threshold, it just fires an action potential. This way it can do summation in space, summation in time. Now we'll see summation in space, like two impulses coming from two different directions on the dendrite can add up and help the neuron to cross the threshold and fires an action potential. Other way possible is Here only we are seeing excitatory. There could be inhibitory inputs also, depending on the neurotransmitter. So let's say a inhibitory neurotransmitter is sent to the postsynaptic neuron, and this inhibits the firing of this neuron or potential of this neuron, whereas this excites. So although uh, first inhibitory just gets the voltage down, but if you get first excitatory and inhibitory, it excites and then inhibitory makes it down. So uh, this these are called the potentials created like this are called postsynaptic potentials, and it could be either excitatory or sorry okay okay it could be either excitatory postsynaptic potential or inhibitory postsynaptic potential, and these potentials can add up in time or can add up in space, and this way the neuron signaling takes place from neuron to neuron, and you have seen what's an action potential. Now coming to again to anatomy, like what are the connections present in the nervous system. So you have some feed forward connections which are just going from input to output directly. These are feed forward connections. You have point to point connections. A point in a brain has a connection to another point, another set of cells in the brain. This is called point to point connections. Then you have convergent connections. That is, you have a region in the brain which is receiving inputs from multiple diverse regions in the brain. So you have multiple regions here and you have converging connections. Diverging connections is from one region, it's just opposite of converging. Just have multiple connections going away from that to diverse multiple regions of the brain. These are divergent connections. Then you have feedback connections where a region is connected to other region, but also the other region is again connected to this region. So this way they have a closed loop communication. This forms a closed loop communication. It's like a closed loop feedback control system, and these are about feedback connections. There is various other types forming loops, reciprocal and reentrancy feedback connections. Okay. And also you have local connectivity at the level of neurons. So here the laminar structure reveals about the local connections. So this is a sheet of cells in your brain. For example, if you take the white matter here, so gray matter, if you cut through this, you'll find these as the laminar structure. This part, the gray matter part, has the laminar structure that is shown here. 1 second. Okay. Yeah. Okay, this is about action. Okay, this laminar structure, each layer has different cytoarchitecture. Like each layer has different types of cells, and each layer has connections from different layers. For example, if you take M1, the primary motor cortex, this is there in, as we discussed, precentral gyrus. This is post central gyrus. So this receives the sensory input and this performs the actions. So you can see how different layers, here the layer 4, it is divided into 6 layers. Layer 4 cells are getting the input, like feedback input from layer 2 and 3 cells of this one. Similarly, you have various projections and connections between these layers, which has inputs, feedbacks, etc. So, and these layers are classified based on different types and basically the cytoarchitecture, the cellular architecture of the gray matter. This is about local connections. Then we have in the lectures we have dealt with an important metric. Like we have seen that size is not a very good metric for measuring anything about the brain, like for example about the intelligent quotient or anything. So scientists came up with a very good metric called encephalization quotient by saying that there exists a power law kind of relationship between the body size and the brain size. So here encephalon means the brain. Encephalon to soma, E by S ratio first. So first they said about encephalon size, about the size of brain, which doesn't give any insights into the uh, any relationship between the brain size and the intelligent quotients or in general intelligence. Then they came up with a more control metric called E by S ratio, and they also found this also doesn't give much information. Then the scientists hypothesized that there could be a power law kind of thing existing between this E and S. So E, they came up with this E is equal to some factor called cephalization factor times S power R. So how they came up with this is they bought an analogy between uh, sphere and surface area of the sphere, volume and surface area. So brain has connections which are on all the surface, like connected to whole surface. So they gave it as surface area and this as volume. So you have it as 2 by 3. That is for one type of hypothesis, one type of shape, but it could be different for different species of animals. Therefore, let's keep that as R, and there is a proportionality constant called cephalization factor. So these relations are measured across different species, and for example you want to know average cephalization across mammals. You calculate that. You have all the classes of mammals that you measured. You have average cephalization factor, and then for a given sample, for example, for a given person, you want to calculate this. So that person has some C, has a cephalization factor, which is equal to E by S power R, right? Yes. So you calculate this for a given person or given animal, and you have average of those species, and you, in order to compare how good they are, you make up a metric called encephalization quotient, which is nothing but individual cephalization factor divided by average factor of that particular group. And this way, this is a better metric compared to just size and size to body size ratio. And this gives some insights, although it might have few conditions where it is not applicable still, but it works better compared to others. So today we have discussed few things from the week one lectures, and this is about it. If you have further questions, you can ask me now. So this is about it. Any further questions? Okay. Uh, cool. It's fine. I think if you have further doubts, you can ask me uh, in the discussion forums. And thank you. That's all for today.